Hello students, in this video lecture we will learn about the meiosis and its different stages. Basically, meiosis is a special type of cell division of germ cells in sexually reproducing organisms that produces the gametes, sperms or egg cells. In meiosis, the chromosomes duplicate and homologous chromosomes exchange genetic information, which is also called chromosomal crossover, during the first division called meiosis 1, and the daughter cells divide again in meiosis 2, splitting up sister chromatids to form haploid gametes. Two gametes fuses during fertilization, forming a diploid cell, which is called zygote, with a complete set of paired chromosomes. Now let's discuss about the stages of meiosis. There are two stages of meiosis, which is meiosis 1 and meiosis 2. Meiosis 1 is also called as reductional division, and meiosis 2 is also called equational division. Meiosis 1 is further divided into four phases. It begins with one diploid parent cell and ends with two haploid daughter cells having the number of chromosomes in each cell. The further stages in meiosis 1 is prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 is divided into four phases, which is prophase 2, metaphase 2, anaphase 2, and telophase 2. And it starts with two haploid parent cells and end with four haploid daughter cells, maintaining the number of chromosomes in each cell. Interphase 1, which is comes in the meiosis 1. In this, nucleus and nucleolus is visible. Prophase 1. Prophase 1 is the longest and most complex phase. Chromatin condenses to form chromosomes. Synapses occur, in which homologous chromosomes come together to form a tetrad, and tetrad is two chromosomes or four chromatids, which is called sister and non sister chromatids. Crossing over of genes occurs in the tetrad. Homologous chromosomes are a pair of chromosomes, one are obtained from each parent. They are similar but not identical. Prophase 1 is further divided into five stages. Number 1, leptotene. Leptotene is the first stage of prophase 1, and all the chromosomes begins to condense, so they become visible as a fine thread, and there is marked increase in the nuclear volume. A process of homology search, which is essential to the initial pairing of homologues, begins during leptotene. Now zygotene. Zygotene stage, also known as zygonema, and this stage begins with the initiation of pairing between homologous chromosomes and it ends with complete pairing. The process of pairing between homologous chromosome is known as synapses, and the synaptonymal complex is formed during this zygote stage. At the completion of zygotene, the paired homologues takes the form of bivalent, and the number of bivalents in each species is equal to the haploid numbers. Now the third stage is pachytene. Pachytene stage is also known as pachynemia, and the process of synapses is complete. The two homologues of each bivalent appears to be attached with each other at one or more points. These attachments are known as chiasmata. Now the fourth stage is diplotene. Diplotene stage is also called diplonema, and the DNA recombination is complete in this stage. The chromatids continue to shorten and thicken, and the four sister chromatids in a group is called a tetrad. The synoptimal complex begins to break down, and the paired chromatid begins to pull apart, causing the strands to separate longitudinally, and the chiasmata tends to become terminalized as the meiotic prophase continues. Now, the last stage is dikinesis. In this, the chromosomes become shorter and thicker due to condensation. Nucleolus and nuclear envelope disappear towards the end of dikinesis, and the spindle apparatus becomes organized, and the centrioles migrate away from one another. Now, Metaphase 1. It is the shortest phase, and the tetrads align on the metaphase plate, and spindle fibers attach to the kinetochores of the centromeres. Anaphase 1. Homologous chromosomes separate and move towards the poles, and the sister chromatids remains attached at their centromeres. Now in telophase 1, each pole now has haploid set of chromosomes. Cytokinesis occurs and two haploid daughter cells are formed. Cytokinesis basically is the physical process of cell division in which the nucleus and cytoplasm divide, giving rise to two cells. Now prophase 2. Prophase 2 is same as prophase in mitosis, and chromatin get condensed to form chromosomes. The spindle fibers are formed and the centrosomes begin to move towards either of the poles. Metaphase 2. In metaphase 2 is same as metaphase in mitosis. The chromosomes align themselves on the metaphase plate, and the spindle fibers attach to the kinetochores on the centromeres. Anaphase 2. Anaphase 2 is same as anaphase in mitosis. Homologous chromosomes do not separate, but sister chromatids separate, and each chromosome is thus broken down into sister chromatids which move towards either of the poles. Telophase 2. Telophase 2 is same as telophase in mitosis. In this, nuclei are formed and cytokinesis is also occurs. Four haploid daughter cells produce, and these cells are called as gametes.